Today I'm going to talk about fertility regulating mechanisms and their manipulation. Seed production is very important for breeders, especially when they are doing some hybridization between different plants to reach good recombination of different alleles. Normally, plants are producing seeds in normal way, but sometimes some of the plants, we know that some of the plant species produce good seeds if they are cross-pollinated, even though they are having high percentage of self-fertilization. The seed setting importance will be more and more if we are crossing two different species, or if we are looking for genes from different species to introduce it to our cultivated plants, or from different genera, because in these cases, in these two we call it interspecific and intergeneric species crosses. In this case, one seed, if it is fertilized seeds and produce good plant, is very important. As I mentioned in the previous slide, fertility between close related species, or when I do crosses with close related species, is very important because I need to transfer certain genes to my plants, and if it doesn't produce freely seeds for the hybrid plants, then I cannot transfer these genes to my population, and I cannot use any I cannot do any recombination of alleles in my breeding program. All these terms of non-producing seeds, or what we call sterility, is coming under different titles. Sometimes we call it infertility, sometimes we call it self-incompatibility. It has many terms that I will talk about it later, but all of these can be resulted from irregularities during sexual reproduction, where during either the production of gametes or during the process of pollen tube growth, or during the fertilization process. And we can summarize these causes of infertility as these irregularities, but not mentioned all of it. I just mentioned examples. For example, malformation of stamen or pistil. Sometimes the stamens or the pistils are malformed, or what we call produced in non-fertile, in non-good shape. Pollens sometimes defects. The pollens is shed and then it is defects, and this has been many times known. Sometimes, for example, in Jordan, they said that this year the production of stone fruits, or the production of certain type of fruits, is not good because the pollen transfer or the pollination came on time where it is the temperature is high or the temperature is low, where the pollens are defects, or ovule aborted sometimes for certain reason, or pollen fail to germinate. Pollen tube is not growing well and cannot penetrate the stigma surface. Pollen tube is not growing in the style. the fertilization sometimes happens, but embryo is not developed. Or before that, during gametogenesis, there is non-pairing during meiosis. All of these from the mother cell development till we end up with a seed, good seed development can affect the infertility system of the. Whatever the cause of this infertility, we as a breeder has to understand it and try to avoid it or to manage it, control it, and get good seed recombination, especially through the crosses between different species and different genera. Infertility can be divided in two types. First of all, incompatibility, and the second is sterility. And we have other related subjects that we want to cover. So in this chapter, we're gonna cover incompatibility systems, sterility systems, different sterility systems, and we will cover some other related subjects. I will start with incompatibility systems. Incompatibility is the case where we have the plant produce normal pollens and normal ovules, but they are not setting any type of seeds due to physiological hindrance that prevent fertilization. It can caused by any mean that can stop the fertilization after pollination, because we produce normal pollens and ovules, but there is something that restricts the self-fertilization or inbreeding, and in this case we are pushed to go to outbreeding and cross-fertilization. There are many families that has, or many species that has this type of incompatibility, which is like in Leguminaceae or Solanaceae or Crucifer or Composite or Gramineae. Many species known in that, like alfalfa, ryegrass, sugar beet, clover, etc. So this phenomena is there. Plants producing normal organelles, but they are not having seed setting. And this happened even in humans. Sometimes couple get married, they have all the normal things, and the physicians told them you are normal, but they are not having any kids. And this is the case. We have two types or two systems of incompatibility. The first one, we call it gametophytic incompatibility, and the second one is the sporophytic incompatibility. Gametophytic incompatibility found in many plant species like potatoes, beets, sugars, tobacco. And this type of incompatibility, the pollen tube growth is controlled by multiple alleles, S1, S2, S3, etc. And we know that the pollen nuclei has one N, which is one allele. So it has S1, S2, S3, S4, S5, etc. While the style tissue is diploid because it is somatic cell, so it has 2N. So what's happened and how this system Let's look to this figure. The stylar tissues that we have different stylar tissue, which is S1, S2, S1, S3, and S3, S4. And the pollen grains that we have that are coming from a plant which has S1, S2. So the pollens that are coming are S1, S2 for all the stigma and the styles. Now, in the first case on the left, the style that has S1, S2, no growth for the pollens. The one that has S1, S3, only S2 is grow and make fertilization. And the one that has S3, S4, both of them are doing growth and they do fertilization. So in this case, no homozygous or no homozygous plants will be produced. So there is no S1, S1 or S2, S2 or S3, S3. All of them will be heterozygous plants. This system of incompatibility is not effective 100%. Sometimes some of the seeds or the stylar tissue allow some alleles to grow, and in this case we call it pseudo self-compatibility. And the amount of selfing in this system is dependent on the environmental conditions and sometimes due to mutations, because mutations can modify the genes and affect the plants where high number of self-compatibility will be produced. Therefore, there is possibility to get some seeds, some self-pollinated seeds in the system, and we can manipulate the temperature to or incorporate what we call modifying genes. So mutation, we don't know, maybe we can do it through mutagenic agents, as I said in mutation breeding, but the environmental condition like temperature can be done, and we can transfer a modifying genes that can increase self-incompatibility. So as I said, self-fertilization happened by pseudo self-compatibility through either playing with the temperature, increasing temperature, or through mutation, and sometimes within the S system, S1, S2, S3, S4, we have one of the alleles, which is called SF, which can do self-fertility, and we call it self-fertility allele, which can be produced by mutations. In this case, if the SF is found in the stylar tissue, or it is coming, it can penetrate and do, it can allow the same allele to come, so we will produce homozygous plants. Of course, sometimes, in addition to pseudo-fertilization and production of SF, we can do polyploidy sometimes, where we duplicate the genes, like in the case of clover, and in this case we are producing a new ploidy plants that doesn't have this incompatibility. Because the stylar tissue is the one responsible of this type of incompatibility, which means that when I show you the figure that the style tissue, for example, has S1 or S2, it prevents any S1 and S2 to come and grow. The style tissue itself is the one that produce the opposition, prevent the growth. So we call it oppositional factor hypothesis, where the S system or the styler tissue prevent the growth of the pollen. And sorry, the number of S alleles that or the number of S alleles that in this system is very large. So we have large number of S alleles that found in or identified in nature. As I said, there is a lot of families that has this opposition factor hypothesis or system, which is the gametophytic incompatibility. One of them is the grasses, and most of the families has one loci responsible, which is the S loci, that's responsible of this type of self-incompatibility, while in grasses we have two loci, S and Z. So we have two different types of alleles that prevent self-fertilization, and this has been described very well in rye and sugar beet. The second incompatibility system is called sporophytic system. The sporophytic system is one locus system, and it is not like the gametophytic where there is possibility for more than one locus. The sporophytic system is a system that has some dominancy, where if S1 is dominant over S2, then all S1 is compatible to S2, and vice versa. S2 will not be compatible. So if we have styler tissue with S1, it prevents S1 and S2. If we have stigma surface with S1 and S2, then this S1 prevent S1 and S2. If we have a stigma with S2, then all S1 can penetrate. So S1 prevent S2, even though they are not the same, while S2 cannot prevent S1, and I can explain it now in the next figure. The other difference between sporophytic and gametophytic is that the hinderness of the gametophytic, as we said before, is found in the styler tissue, while for the sporophytic, it is found in the stigma surface. I will explain the sporophytic system here on this figure. The first, on the left side of the figure, you can see that we have a S1, S2 plant, and we receive two S1 and S2 pollens. Now, as we said, S1 is dominant over S2, so now S1 is preventing S1 and S2, preventing S1 and S2. In the second figure, we have S1, S3, and we receive the same type of pollens. Now you can see, if we are talking about gametophytic, then S2 should go and penetrate and make fertilization. But because S1 is dominant over S2, it prevents S2 from penetrating. It will not, so S2 is not going to be penetrating because S1 is oppositing the S2 because it is dominant over it. While if we have here S2 instead S2 S3, then now the S1 and S1 can penetrate. Okay, and if we have now stigma of S3 S4, then S1 and S2 can penetrate like the gametophyte. So here you can see that S1 preventing S2 from penetrating because it is dominant over. The sporophytic system is found in dicotyledons, but it's not found in monocotyledons. Now I can summarize the difference between the two systems. First of all, the gametophytic can have more than one loci. Sporophytic only one loci. The gametophytic doesn't have any dominancy. Sporophytic has dominancy. The opposition in the gametophytic is found in the stylar tissue, while the opposition in the prevention in the sporophytic is found in the stigma tissue. Even though we have this type of incompatibility that found or prevent the growth of the pollens in the stigma or in the style, there are many practices that breeders were doing to overcome the incompatibility, especially sporophytic incompatibility, because we said sporophytic incompatibility found in the stigma surface. So many methods has been used, like bud pollination, rupturing the stigma surface, grafting, which is very well known in many cucurbitaceae. Sometimes we do electrical shock to the surface of the stigma, or increasing the CO2 concentration. And all of you know that the CO2 information is used to increase the fruit setting in many cucurbitaceae plants, especially in the greenhouse. They are used to increase the fruit setting of the plants. So it helps also on overcoming the sporophytic incompatibility. In the beginning of the chapter, I told you that we like to understand incompatibility and utilize it in our breeding program, or manage it in order to be utilized very well, even though it prevents self-fertilization, but we can use it to increase our outcross or cross-pollination, and it can be used to produce hybrids. For example, in many vegetatively propagated plants, self-incompatibility was used for doing some crosses in order to produce hybrid seeds. We use it in single, double, and triple crosses of brassica, which I will explain what we mean by single, double, and triple crosses later. Or if I need pseudo self-incompatibility and it is needed very well, then I can use either SF by introducing this allele, or I can increase the number or the percentage of pseudo self incompatibility and plant breeding, which means how we can utilize the incompatibility in our breeding programs to facilitate self-pollination and produce inbreds in self-incompatibility systems. We know that this system is preventing self-pollination. As I mentioned in the previous slide, we can use easily either pseudo-incompatibility or SF or self-fertilization alleles. Now, in case of using this for outcrossing, it is used widely in facilitating the hybrid seed production. And in this case, we don't need to control pollination because the plants themselves are self-incompatible. It has self-incompatibility system, which means that it doesn't produce self seeds. And in this case, if you want to utilize it, it has to be completely is incompatible, where 100% incompatibility is found. This system has been used in producing hybrids from self-pollinated crops through self-incompatibility clones. And this can be clear in the coming slides, where we can show you different types of crosses that has been utilized to produce hybrid seeds. As I mentioned previously, we use this system to produce single, double, and triple crosses in vegetatively propagated crops. For example, the example here is cabbage or kale or Swedes or in brassica. The first type of cross that we can use, we call it single cross. Single cross is crossing two inbred lines to produce a hybrid plant like S1, S1 with S2, S2 to produce S1, S2, and this is called single cross system. In this cross system, we need to have many crosses because usually you wouldn't have a large amount of seeds to produce. So commercially, we need to produce a lot, or sometimes we use another means like bud pollination for commercial seed production. To overcome the lack of seed production, sometimes we do double cross. We call it double cross, which produce more seeds than the single cross. The double cross required that we have four inbred lines like S1, S1 with S2, S2, and S2, S3, S3 with S4, S4, and we produce the first hybrid S1, S2 and the second hybrid S3, S4, which is two single hybrids. Then we cross the two single hybrids to produce many single hybrids. In this case, we call it double cross system. Or as in the second, now we'll go to the triple cross system, which was proposed to increase the number of seeds more and more, and it involves the 6 types of inbreds. And we'll start with the first cross, which is S1 by S1 by S2, S2. And then the single hybrid produced S1, S2 will be crossed with S3, S3, and resulted in 2 types of hybrids, S1, S3, and S2, S3. In the other side, we will have S1, S4, S4 by S5, S5, which produce S4, S5. Then we cross it with S6, S6, and produce S4, S6, S5, S6, and then we recross again S1, S3, S2, S3 from one side with S4, S6, and S5, S6 in the other side to produce the hybrid that we need. And in this case, we are expecting to produce, generally we produce more and more seeds. By this, we are ending part 1 of chapter 7, and I will go to part 2.